Welcome to Brain Gauge. I'm Mark Tarmadol, and today we're going to talk about amplitude discrimination. That's a score, or the test that you use to get your accuracy score, and sometimes you use it to get a plasticity score. Anyway, we're going to talk a little bit about the mechanisms that are involved in amplitude discrimination, and this is a test where you answer the question, which one is larger? It's a little more complicated than that, but I'm going to try to go through a little bit of it. So when you look at this slide, what we're trying to talk about here is notice down in the two boxes below. Basically, what I want you to look at are the two squares where you have a short stimulus that's being compared to a long stimulus. And those are the different stimuli on the skin. And what we're doing is comparing a short versus a long stimulus in terms of what's the percept. And how does that percept feel when you deliver a long repetitive stimulus versus a very short stimulus? And what you notice is that it gets very sharp. And this is just like if you put a stimulus on your finger and did it over and over and over, it would get more and more sharp and you could localize it better and better. Well, one of the things that we learned over the last 40, 50 years and brain studies, and I was part of that for about 35 years, was looking at cortical activity. And the cortical activity actually parallels the percept. So in the top right-hand corner, what you see is an actual pattern of cortical activity that's taken in response to a repetitive stimulation. So notice the peak activity, the red spot that you see. Well, let's see if we can put a circle around it. You notice that, and that red dot, that peak, it's surrounded by an area of inhibition. And so when you deliver long repetitive stimulation, you get this nice peak, and basically everybody, all the neurons around the most active neuron get much weaker. That's a lateral inhibition process. And so basically, if you look in the graph here, below that, it's showing a difference between a short duration stimulus and a long duration stimulus. Now, you guys, when you do your cortical metrics test, when you use the brain gauge, you're basically not really aware of differences in short versus long stimuli. The reason this is here is to try to display the actual mechanism of what's going on in terms of lateral inhibition. And basically these are the mechanisms that go on very, very rapidly. And the stimuli that are delivered to the skin during the cortical metrics test is really 500 milliseconds. So it's really not that long, but a lot happens in that amount of time. Let's go on to the next slide. Now suppose we deliver stimuli to two adjacent fingertips. So basically they are activating two places in the brain that are side by side. And let's see, what would be the percept there? Well, under a healthy condition, when you have a fully intact functional connectivity, you get something here. On the left, you get two nice, highly resolved peaks, so you can really tell the difference. Now, in the case on the right, you have two peaks that are not very resolved, and that's because the connectivity between those areas is disrupted. So basically, it's really important that with repetitive stimulation, that you actually get those, you actually get the carving out and the funneling between the two stimuli. So how do we actually measure how well you can carve out the two different stimuli that are side by side? Well, what we do is we use a tracking protocol. And tracking is really just like reading an eye chart. It starts, when you read an eye chart, you start out reading a large letter, and they get progressively smaller, and at some point you can't read anymore. Well, the way we do amplitude discrimination is that we deliver two stimuli, and let's say right here, you get a large stimulus on the left, and here you get a small stimulus on the right. And the first time, you might compare, let's just give it some random numbers, 200 versus 400. And you can easily tell the difference between those two. Well, then the next test, you compare 200 versus 360. And the next test, you compare 220 versus 340. So, in other words, the difference between the two keeps getting smaller. So the difference starts out here and it keeps going down and down and down until you start tracking. You get to the point where you're just guessing. And that's the whole point. You should start guessing at some point, and usually you guess, and that's called your difference Lyman, or your DL. If you look at your raw scores, on your analysis package, that's what is reported for amplitude discrimination, is your DL. Now, if you look down here on the bottom, you see DLs of, basically for control, for migraine, and for concussion. And those are just typical disruptions have occurred in those different neurological disorders. And in those cases, their DLs are higher. So the smaller your DL, the better. The smaller your difference Lyman, or your amplitude discrimination score, the better it is. And the smaller your amplitude discrimination score is, the higher your accuracy score is. So your accuracy scores get better as your amplitude discrimination gets lower. Okay, so looking at amplitude discrimination levels, and you just kind of start wondering, well, how could anything really change amplitude discrimination metric, and how does that relate to what's going on in the brain? And if you look at this slide, basically what we're doing is we're comparing two different conditions. One is a pre-drug and one is a post-drug. The pre-drug is normal condition, and basically notice the two X marks on the left that are activating. Those are locations of electrical stimulation for different areas of basically columnar organization, two columns of organization, or basically, and note that those groups of cells get active, and the histogram below it is very well separated. Now, post-drug, those cells, you look at the histogram on the right, and it's no longer separated. But you see that those those two areas of activation have now merged, and you can't distinguish them. So the drug that we gave in this is a very mild GABA antagonist. It was 10 to the minus 6 molar bicuculline. And basically it turns off inhibition. And inhibition plays a huge role, and it's part of functional connectivity. So if you turn off a really important component of functional connectivity, then the brain doesn't work as well, and then amplitude discrimination is simply not going to work as well. Now, one thing that you might have noticed is that when I described the amplitude discrimination task a minute ago, I was describing simultaneous amplitude discrimination. In other words, we were delivering stimuli to both fingers at the exact same time. Yet, when we do the cortical metrics test, we do two ways. We deliver that in two different ways. We do the simultaneous amplitude discrimination I just described, and we do what we call sequential. That's where we deliver one stimulus to one finger and one to the next. Now, if you look at this slide and imagine, well, delivering simultaneous amplitude discrimination, it won't make a big difference under those two conditions. Yet, what we find is sequential amplitude discrimination does not have as big effect because lateral inhibition doesn't come to play as much. And the reason we do that is so that we can compare the two metrics. That's the way we can get a measure of lateral inhibition on each individual by comparing their simultaneous to their sequential amplitude discrimination. So it's worth noting differences between centrally and peripherally mediated measures. Okay, so this is simply because this is a place to talk about amplitude discrimination. And what you see on the left is over here on the left, you see the amplitude discrimination plot. And what it's showing is that basically amplitude discrimination stays very constant for all age groups as long as you're healthy. So that's very different from traditional sensory testing, which says you get worse as you get older. The graph on the right, where higher numbers are worse, thresholds go up, that means sensitivity goes down. That means basically that's a traditional measure of sensory testing where people look at sensory threshold or sensitivity, and it's really more a reflection of skin physiology than it is of the central nervous system. So central nervous system mechanisms actually stay pretty well intact as long as you stay healthy. All right, so let's go to another concept. Let's talk about illusory stimuli. If you look at the left, and the question is, how many triangles were drawn? And you look at that very closely and you say, you might count a certain number of triangles, but the answer is there are no triangles drawn. What is actually there is an illusion, and the brain actually connects the dots so that you actually do see triangles. Now, on the right-hand side is a different illusion, and let's ask the question, which center circle is larger? Well, if you're just looking at it, you probably would say the center circle on the left is much larger. Well, that is what would be called a context-dependent illusion. In other words, the circle in the middle is evaluated by your brain in the context of how large the other stimuli are. We can do the same thing with a tactile illusion. The difference being it's much easier to measure the result of a tactile illusion. So let's see what we can do. Okay, let's look what happens when we modify the amplitude discrimination task. Now, remember, with the amplitude discrimination task, we're just asking which of two stimuli are larger. And in this case, we are, we're doing the exact same task, and that is being done over here. But before we do that, we're doing some illusory conditioning. What we do is we stimulate one of the fingertips, one of the digit tips, and what that does is that makes that stimulus, this stimulus here is going to make this stimulus feel much, much smaller. Now, when you make that stimulus feel smaller, it's going to be much harder to tell the difference between the large stimulus and the small stimulus. So what we did, basically we deliver this illusion on both sides so that you get a couple of different repeats. But basically, what happens when you deliver that illusory conditioning is you make the tracking much worse because you no longer can tell which of the two stimuli is larger. You have a lot of trouble with that. And typically, the way we have it set up, it makes people, much worse, 30 to 40 to 50% worse. Now, let's look at the DLs for these different tests. And if you ever take this cortical metric, you'll see the abbreviation SSA, or single site adaptation. And what you see in the graph here is you see controls, migraine, and concussion. And in each case, you see the amplitude discrimination task, you see the DL for it in blue, and in purple, what you see is is a single site adaptation DL. That's a different difference lineman. All of a sudden, those are usually, like for control, it's much bigger. But the key factor is the green arrow. That is the adaptation metric. What is the difference between those two? And so obviously for controls, there's a big difference. It makes you much worse. For migraine, it doesn't make you much worse. For concussion, it doesn't make you much worse. And that's because adaptation has been disrupted. So not only are you worse at the amplitude discrimination task, but now when you do this conditioning stimulus, it doesn't make you much worse. So there's a lot of advantage of this is that it makes it difficult to cheat, which is not always a problem, but for home users. But it is sometimes a problem with people that are trying to feign injury. We've seen the adaptation metric to be sensitive in a number of different conditions, and are all published. You can go read those publications on the website on our publications page. And you want to see that's on corticalmetrics.com/publications. And that's probably enough for today. Thanks for listening.